Hello everyone, I'm Yawen Zheng from Crown Bioscience. And today I'm very glad to have a chance to share what I'm doing in Crown Bioscience, the preclinical histopathology image analysis using the deep learning models. Here is the agenda. First, I will give some details about our background, and then I will introduce our project. And the last step is the future directions. Okay, here's the background. We know that variations in factors like the blood vessel density, fibrosis ratio, and necrotic areas within the preclinical histopathology images are usually considered as critical markers for assessing the efficacy of the oncology treatments, like the slides shown here. And the general way to analyze the pathology image is that the pathologist use the mouse to manually outline the required regions, like the video shown here. And this process is very time intensive, and it relies heavily on the expert judgment, and the results are sometimes inaccurate and biased. So we started to think that are there any more efficient and accurate methods to pinpoint the desired sections? And in fact, we're inspired by the deep learning developments in the computer vision. And here we take the general image as an example. For the left image showing here, if we put this image into the deep learning model, the output of the model may be the horse. We call the horse as a label. It means all pixels is assigned with a label horse. And this task is called classification task. And for the model image, the deep model may think that all pixels and all horses in this image are different from each other. So the white horse is marked by the brown color, and the nearby horse is marked by the green color. And this task is called instance segmentation task. But for the right image, the model thinks that the pixels belong to the horses are the same with each other, but they are different from the pixels belonging to the sky. So the horses are marked by the yellow color, and the sky is marked by the red color. And this task is called semantic segmentation task. And the classification and segmentation task is to assign the label with each pixel in the image. And we believe that the segmentation and the classification task are pretty similar to our project, because in our project we want to use the deep model to assign the pixels in the preclinical histopathology image with a label information, and which can make the parts in this image are marked by the different colors, are marked by different colors, right? And luckily, there are some existing studies about how to use the deep learning models to analyze the histopathology images. For the academic aspect, the Mammoth lab is one of the classic labs. That this lab is supported by the Harvard Medical School. And the image showing here is one of their classical research. And the input of this deep model is the WSI image, and this image has very different resolutions. And the first step is you need to choose the appropriate resolution, and after that, we call it human star level annotation step. And in this step, they let the expert to outline their regions that maybe belong to the tumor. So if the image with the blue line and this image is marked by the tumor label, and if this image without the blue line and this label is marked by the no tumor label. And the next step is patch extraction, because sometimes we choose the property resolution. The size of the image is pretty large, so we need to cut the image into patches with the same size. And after that, we need to import these patches into the model, because this model uses the annotation information, but this annotation is size level instead of pixel level. So it's called weakly supervised classification task. And for the industry aspect, the Hello software provides a very powerful tool for the pathologists to use the mouse to outline the regions they are interested in. And maybe they wait for several minutes, and they can get the segmented results showing here. And the different color means different semantics. For example, the green means the stroma, and the red means the tumor. Although there are some amazing research and studies here, they still have some limitations. For example, like the deep model embedded software is often very expensive, and they may not satisfy all the needs that the pathologists need, such as the interactive sorting function. And the second is that there are some deep models that can be independent of the annotation information, but they have a large parameters that can make them very hard to be trained. And the third is that several models can have the can be dependent with the manual annotation information that can achieve very higher accuracy. But like I said before, the whole process is labor intensive and time consuming. So based on this limitations, we propose our goal. We want to develop a deep model that can be independent of the annotation information. And meanwhile, this model can quickly and accurately segment the histopathology images. And the second is that we hope that our platform, our deep model, can let the pathologists have a chance to define or specify the number of semantics, like the number of paths that they are interested in. The third is that we want to embed our deep model into an interactive and user-friendly platform, and this platform can make the pathologists manually modify the parts that they need. And the second is that we want the platform that can automatically generate the Excel-based reports for the pathologists for the analysis for the further analysis. So here is our workflow. The left image is the raw data. They are WSI images, and we want to import the WSI images into the model, and then we can get the segmented results. Then in the red color, images shown here, different color means different semantics. For example, the red means the tumor, and the orange means the necrotic areas, and the green color represents the red cells. So the critical step for our deep model is to generate reliable masks. And here is the details. Like I said before, the WS image has very different resolutions. So the first step is to choose the appropriate resolution, and the next step is contouring step. Why we need that? This is because that the background the background occupies most parts of the WSI image. We want our platform to focus on the tumor area instead of the background. So we use the machine learning based algorithms to get the areas that we are really interested in. For example, the areas between the green and the blue lines are the parts that we need. The next step is the patch extraction. The patch size is 192 multiplies 192. And then we import the patches into our deep model. The backbone of our deep model is the pre-trained ResNet 15. The reason why we use this model is that this model is one of the very classical models in the classification tasks, and this model has been well trained by the ImageNet dataset, and this dataset is an authority image dataset. And the last is that it has very unique function. It's called residual function, and with this function, the model can be very deep. We know that the deeper the model can have more chances to learn representative and fine-grained information. And the difference between our model and the ResNet 50 is that we drop the last block, and it means that we drop the last block, and the output of the average pooling is what we need, and this output is called hidden features. Because we want our deep model can be independent of the human annotation. So the next step is that we need to assign the label to each pixel with the unsupervised algorithms. Which means that if we assign the label to each hidden feature, the patches can be assigned with a label, and all the pixels in one patch can have the same label. So once we get the hidden feature, the next step is we use the k-means algorithm to let the feature has a label. And after that, given the control information, we use the visualized algorithm to get the marks that we need. And then we can have the segmented results. We have embedded our platform into a platform, and this platform has been tested and developed into beta version. And this platform has two sections here. The first section is segmentation section. And in this section, you can adjust 3 parameters, and then you can immediately get the areas that you are interested in. And if you submit this operation, you can wait for a few minutes. You can get the segmented results, like the modification present here. The green color means the necrotic areas. The orange color means the tumor itself. Like we said, we have the modification section. And in this section, the pathologist can use the mouse to outline the parts that they think the parts are colored by marked by the wrong color. So after this operation, they can get the modified results shown here. And here is the performance. The above is the original image, and the below is the visualized results. For example, in these 6 circles, the necrotic areas are marked by the same color, the green color, and the tumor are colored by the orange color. It means that our platform can distinguish the necrotic and the tumor areas. But for the pixels in these 4 circles, the pixels are supposed to be labeled as the same color, right? But they are marked by the different colors. So it means our platform still has the room to be improved. But for the middle image, we can see our platform can distinguish the necrotic and tumor areas very well. And for the last image, we can see that besides the necrotic and tumor areas, our platform can also detect the red blood cells. And besides the segmentation and modification sections, we have another section. And this section can automatically detect blood vessels and fibrosis areas. If we post the original image into our platform, the platform can give the visualized results. And in the visualized images, all pixels belong to the blood vessels are marked by the green color. And sometimes the pathologists may be interested in specified regions instead of the whole image. So we allow the experts to use the mouse to outline the regions that they are interested in. Then our platform can only focus on the specified regions and to do the further analysis. And here is our future work. Actually, we are inspired by the segment anything model. We call it SAM model. And the aim of this model is to call out any object in any image. So actually, we have done some experiments on single patch. And here is the example, and we can see that SAM can accurately segment the large area components. The green, the blue color means the tumor, and the baby blue color means the background, and the green color maybe means the necrotic areas. And besides it, we found that the SAM has the potential to segment the relatively small area components, especially the cells. So we map the single patch into the whole image. We can see that the SAM does not perform well. We assume that this is because there is no interaction between the patches. So the next step is that how we let the patches communicate with each other, and we believe that if we can achieve this, the performance of the SAM can be improved a lot on our image. So that's all. Thanks for your listening. And if you have any question, you can ask me. Okay, thank you.